Hello and welcome to our next lecture on enzyme and coenzyme. Myself, Dr. Akhilesh Kumar Mishra, Professor, School of Pharmacy, BBD University. Here is the general characteristic of enzymes. Enzymes are usually a protein and catalyst acting as a catalyst for a specific biochemical reactions. Each cell in the human body contains near about 1000 of different enzymes, and every enzyme in cell requires its own specific enzymes. Mean a specific reaction will take place in presence of a specific enzymes. Most enzymes are globular proteins. A few enzymes are made up of RNA which catalyzes reaction involving nucleic acids. Enzymes undergoes all the reaction of proteins, and enzyme denaturation due to pH or temperature changes their conformation. A person suffering from high fever run the risk of denaturation of certain enzymes. Next is the enzyme structure. There are two type of enzymes: simple enzyme and conjugated enzymes. Simple enzyme is only composed of proteins, and conjugated enzymes have two parts. First one is apoenzyme, and other one is cofactor or coenzymes. Apoenzyme is a conjugated enzyme without its cofactor, and it is the proteinous part of the enzymes. Coenzymes, also known as cofactors, this is the non-proteinous part of the conjugated enzyme. This represents the apoenzyme, and this represents the coenzyme. These apoenzyme and coenzymes conjugate and form the conjugated enzymes complex. In the coenzymes, metal ion often act as a additional cofactors like zinc ion, magnesium ion, manganese ion, and uh, ferrous ion. A metal ion cofactor can be bound directly to the enzyme or to the coenzymes. Coenzyme is a small organic molecule acting as a cofactor in a conjugated enzymes. Coenzyme are derived from vitamin or vitamin derivative. Many vitamins act as a coenzyme, especially B class of vitamins. These are the term and their definition. Enzyme are defined as proteins, only enzyme that facilitate a chemical reaction. Coenzyme is defined as compound derived from a vitamin, example NAD+, that assist an enzyme in facilitating a chemical reaction. Cofactors are metal ions that are assist an enzyme in facilitating a chemical reaction. Apoenzymes, proteins only part of enzymes, that is the proteinous part of the enzymes. For example, isocitrate dehydrogenase that require an additional coenzyme to facilitate a chemical reaction, not functional alone. Polyenzymes are combination of the apoenzymes and coenzymes which together facilitate a chemical reaction. Next one is enzyme nomenclature. Enzyme are named according to the type of reaction they are participated or catalyzes, or it depends on the substrate where they act. Suffix ase is added. For example, lactase, amylase, lipase, or proteases denotes an enzymes. Some digestive enzymes have the suffix in. For example, pepsin, trypsin, and chymotrypsin. These enzymes were the first one to be studied. Next one is prefix denote the type of reaction the enzyme catalyzes. For example, oxidase, redox reactions, hydrolysis means addition of water to break one component into two parts. Substrate identity is often used together with the reaction type. For example, pyruvate carboxylase and lactate dehydrogenase. Here is the some example, the enzyme class and reaction, which type of reaction they catalyzes. For example, oxidoreductase enzymes. This catalyzes redox reactions. Example is dehydrogenase. Next class of enzyme is transferase. This transfer a functional group from one molecule to another. For example, transaminase. Hydrolysis enzyme, which shows hydrolysis reaction. For example, lipases and proteases. Next class is lyases enzyme, addition and removal of atoms from double bonded, and for example is decarboxylases. Next class is isomerase. Isomerase, this shows isomerase reactions, and name of the enzyme is isomerase and mutases. Next class is ligases, and these shows synthesis reactions are joining up two molecule into one. For example, synthetases. link to a smaller molecule and form a larger one. Next is the enzyme active site. The active site is the specific portion of enzymes where the substrate bind while it undergoes a chemical reaction. The active site is a 3D services like cavity formed by secondary or tertiary structure of the protein part of the enzymes. The enzyme can have more than only one active site. The amino acids or side chains in the active site are important for determining the specificity of the substrate. Next is enzyme substrate complex formation, where the substrate bind to the enzyme's active sites and form an enzyme substrate complex. This is the active site of the active site of the enzymes, and here is the substrate, where they react to each other and form an enzyme substrate complex. And at last, the product and the enzymes are removed from each other. Lock and key model of enzyme actions. The active in lock and key model, the active site is fixed with a rigid shape like a lock, and the substrate to function as a key must fit exactly into the rigid enzymes. Upon completion of the reaction, the product are released from the active site, so the next substrate molecule can bind to the active site of the enzymes. Here is the substrate and enzyme active sites, and substrate fit into the active site of the enzyme, and at last product are released from the active site of the enzyme. Next one is induced feed model of enzymatic action. Many enzymes are flexible and constantly change their shape. The shape of the active site changes to accept and accommodate the substrate. Conformation change in the enzyme active site to allow the substrate to bind. And here is the substrate, and here is the active enzymatic active sites. And after the complex formation, the product is released from the enzyme substrate complex. Enzyme specificity. There is a many type of specificity of a enzyme. A specific type of enzyme catalyzes the specific substrate. First one is linkage specificity. The enzyme will act on a particular type of chemical bond irrespective of the molecular structure. For example, phosphatases hydrolyzed only phosphate ester bond, and chymotrypsin catalyzes the hydrolysis of peptide bond. The next type of specificity is stereochemical. The enzyme can distinguish between stereoisomer. For example, L-amino acid oxidase catalyzes only N-amino acid but not of D-amino acid. There are many factors which affect the enzyme activity, and there are mainly four factors affecting enzyme activity. First one is temperature, next one is pH, third one is substrate concentration, and fourth one is enzyme concentration. First factor is temperature. With increase in temperature, the kinetic energy increases, by which collision increases and increased reaction rate. Optimum temperature at which enzyme shows maximum activity is 37 degrees centigrade, and when the temperature increases beyond the optimum temperature, there is change in the structures of the tertiary protein of an enzyme, and leads to the denaturing. For example, in case of fever, some enzymes becomes denatured. Little activity is observed. Next factor is the pH. Optimum pH is the pH at which the enzyme exhibit maximum activity. Most enzymes are active over a very narrow pH range. A small change in pH can result in enzyme denaturation or loss of their functions. And maximum enzyme is active at physiological pH range of 7 to 7.5. But digestive enzymes are an exception. For example, pepsin, which is found in the stomach, is active at 2 pH, and trypsin is active at the 8 pH. And next factor is the substrate concentration. When we increase the concentration of the substrate by keeping the enzyme concentration at a constant, the rate of reaction increases until the saturation point is met. At saturation, the reaction rate stays the same even if the substrate is increased. Here we can say as the concentration of the substrate increases at a point, the reaction is in, the rate of reaction is increases, and upon the saturation, with a constant rate, the reaction is increases. The incoming substrate molecule must wait for their next factor is enzyme concentration. As the substrate is kept constant and the enzyme concentration is increased, the rate of the reaction is also increases. Here we can obtain a straight line. In summary, we can see the mechanism of enzyme actions. First mechanism in lock and key model, and second mechanism is induced fit model. And here is the factor that affect the rate of the enzymatic action. First one is temperature, and second one is pH. Next is concentration of the substance, and last is the concentration of the enzyme. Enzyme inhibition. There are three types of enzyme inhibition: reversible competitive inhibition, reversible non-competitive inhibition, and reversible inhibition. uh, reversible competitive inhibition, the competitive inhibitor resemble to the structure of the substrate, and it bind with the active site of the enzymes by which the normal substrate cannot bind with the enzymes. Many drugs may act as a competitive inhibitor, like antihistamines inhibit histidine dicarboxylases, which convert histidine into histamine. Reversible non-competitive inhibition. The non-competitive inhibitor decreases the enzyme activity by binding to the site on the enzyme other than the active sites, by which the substrate cannot fit into the active site of the enzymes. For example, heavy metals like lead and mercury bind to the SH of the cysteine away from the active site, which disrupt the tertiary structure of the enzyme. Next is irreversible inhibition. In irreversible inhibition, the inhibitor inactivate an enzyme by binding to its active site by a strong covalent bond. Irreversible inhibitor do not resemble to the substrate. Addition of a excess substrate does not reverse the process and cannot be reversed. Uh, for example, chemical warfare like narrow gases and organophosphate insecticides, which is act as an irreversible inhibitor of an enzyme. Here we can see in a picture the type of enzyme inhibitor. First one is competitive enzyme inhibitor, next one is non-competitive, and last one is irreversible enzyme inhibitors. Uh, Next topic is allosteric enzymes. Allosteric enzyme has a quaternary structure, have two sites for the bonding. First one is bonding site for the substrate, which is known as active site, and the second binding site for a regulator molecules, which is known as a regulatory sites. Active or regulatory binding sites are distinct from each other in shape and location. Here is the different effect of a positive and negative regulator on a allosteric regulations. When a negative allosteric regulator are bind to the allosteric site of the enzymes, the shape of the active sites change by which substrate cannot be bind. While in case of positive allosteric regulator, the allosteric regulators bind with the allosteric site of the enzymes, and the site for the specific substrate is available for the binding of the substrate. And here is the feedback control of the enzymatic reaction. In feedback control, the reaction sequence is controlled by the product of the reaction. Here is the A substrate changes into B, and B changes into C, and C is changes into D, and D is the main product of the reaction. These D main product of the reactions send the feedback control to the enzyme 1, by which reaction can be stopped. Vitamins as a coenzyme. Many enzymes require B vitamin as a coenzyme and allow the enzyme to function. Coenzymes serve as a temporary carrier of atom or functional group. Coenzymes provide chemical reactivity that are apoenzymes lacks. Vitamin B does not remain permanently bonded to the apoenzyme. After the catalytic action, the vitamin is released and can be repeatedly used by various enzymes. The recycling reduces the need of large amount of vitamin. Here is the some drugs which used as a inhibiting of enzyme in activity. For example, ACE inhibitors it inhibit angiotensin converting enzymes and lower the blood pressure. Sulfa drugs, which is antibiotics acting as a competitive inhibitor of bacterial enzymes or stop the conversion of PABA to folic acids. And next is penicillins, beta-lactam antibiotics, and inhibit the transpeptidases. And the transpeptidase enzyme strengthens the cell wall of the bacteria and forms peptide cross-link between the polysaccharide strand in bacterial cell wall. Without transpeptidase enzymes, the cell wall becomes weak. Here is the medical uses of the enzymes. Some enzymes are used in the diagnosis of the disease. For example, lactate dehydrogenase is normally not found in high level in blood as it produces in the cells. Increased level of LDH in the blood indicate the myocardial infarction or heart attack. Tissue plasminogen activator TPA activates the enzyme plasminogen that dissolve blood clots used in the treatment of myocardial infarction. There is no direct test to measure urea in the blood. Urease convert urea into ammonia, which is easily measured and used as urea indicators. And blood urea nitrogen is used to measure kidney function, also kidney function test. High urea level in the blood indicate kidney malfunction. Next is isoenzymes. Isoenzyme catalyzes the same reaction in different tissue of the body. For example, lactate dehydrogenase LDH consists of 5 isoenzymes. LDH1 is a isoenzyme is more prevalent in heart muscles, and LDH5 is found in skeletal muscles and liver. Isoenzyme can be used to identify the damage that disease organ or. Here is the some enzymes which are assayed during the diagnostic medicine. Example is lactate dehydrogenase enzymes used in the diagnosis of heart disease and liver disease. Creatinine phosphokinase used in the diagnosis of heart diseases. Aspartate transaminase is also used in the disease of diagnostic of disease of heart, liver, and muscles damage. Alanine transaminase is also used to diagnose the heart diseases, liver diseases, and muscle damage. As GGTP is also used in heart disease and liver diseases. Our alkaline phosphatase ALP is used in the bone in the diagnosis of bone diseases and liver diseases. So this is the all about the enzymes. Thank you for your listening.